Okay, so good evening, everyone. So I welcome you all to the week 10 tutorial session of the NPTEL course Genetic Engineering, Theory and Application. So in week 10, there are a lot of concepts that have been discussed. So um, we will discuss each of them one by one. Uh, so so there will be an extra session around on 12th of October. You will get the notification. So what I'll do today is I'll not discuss all the spectroscopic techniques that have been discussed. We will go one by one. We will first try to understand the two spectroscopic in just the UV visible spectroscopy. We will try to understand the basic of what is spectroscopy, and in the extra class or maybe in the next session, we can talk about the MALDI TOF mass spectrometry method and the IR spectroscopy. Okay. Uh, okay. Okay. So I'll start today's session. So let's share today's slides. Okay. So I hope the screen is visible. Hello everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRFTS for this NPTEL course, Genetic Engineering, Theory and Application, and this course is being run by Professor Vishal Trivedi of IIT Guwahati. Let's first have a quick recap of what all we learned in week 10. Week 10 is a continuation of what we started understanding in week 9. Yeah, what all we So just to recap, so once you have purified your protein, for example, now the next step is to understand whether the protein that you have purified is the correct protein. So we go for different characterization. Structural, we can go for sequence characterization. We can go for structural characterization. We can go for functional characterization. So there are different methods by which we can analyze whether the protein which we have purified is the correct protein or not. Okay. So last class we understood, we discussed about the electrophoretic technique how we can analyze the protein by running SDS page gel. What is the principle of SDS page, and and then also we learned about the Western blotting technique. Uh, so. Uh, Uh, that is one way of characterizing the protein, but when you are running a Western or when you are running a gel, um, that will not give you the exact molecular weight of your protein. So it is a more you can say it is a qualitative method of characterizing the protein, but it will not give any information about the sequence of protein, right? What is the sequence of the protein, or um, what is the behavior of the protein? the behavior of a protein will depend on what are the functional groups which are present on a in a protein, what are the side chains that are present. So utilize or what are the aromatic amino acid residues that are present in your protein. So uh, all these residues or all these characters in a protein can be analyzed by using several spectroscopic methods. Okay, so we can utilize spectroscopy to understand the behavior. Also, these are basically analytical techniques by which you can characterize your protein, and uh, you, you can understand how the behavior of a protein using different kinds of spectroscopic methods. You can utilize UV visible spectroscopy, you can utilize fluorescence spectroscopy method, or you can also go for CD spectroscopy for understanding the secondary structure of a protein. So we will learn all the spectroscopic techniques one by one. So today we will only discuss the sequencing method and the UV visible spectroscopy UV visible spectroscopy method for analyzing or characterizing your protein of interest. So not only protein, we can have also DNA, other biomolecules also can be characterized using the spectroscopic techniques, but mostly these techniques in biological um, in most of the labs which are working on biological macromolecules, proteins are mainly used for the characterization. Uh, so this is where we started in the last session that if we know about the molecular weight of a protein, then we can characterize our protein. So we can understand if the protein that we have purified has the correct molecular weight or not by doing these techniques by gel filtration chromatography or by electrophoresis. So we how we can use gel filtration chromatography for characterizing the protein based on its molecular weight. So your gel filtration chromatography will give you a profile where you will have the absorbance at 280 nanometer on the y-axis and the volume eluted on the x-axis, and depending on where your protein is getting eluted, you can get the molecular weight of your protein. So, but for getting the molecular weight, you need to run several standards. So, you need to run several standard proteins for which you will get the you will have a standard curve where you will get the molecular weight of the protein and the volume eluted. Now, once you have the standard curve, then depending on the protein of interest, your protein where it is getting eluted, you can extrapolate that and get the molecular weight of your protein. And also electrophoresis, we discussed about the principle of SDS page. Now, another way of characterizing our protein is by the with the help of sequence. So you all know that the protein is a what? Protein is a biomacromolecule which is made up of amino acid residues. So the amino acid residues can be considered as the monomers of protein, and there are 20 different types of amino acid residues. So these amino acids are linked to one another through the peptide bond with the help of peptide bond. So so each protein has a character or has a unique amino acid sequence which can be utilized for understanding whether the or for analyzing our protein and characterizing the protein as well. Okay. So how we can characterize the protein based on its sequence. So what is done is that in the traditional scheme for sequencing large proteins, the amino terminal amino acid residue was first labeled and its identity determined. So when you have a protein sequence, when you have a protein has an N-terminal end. So the N-terminal end will have a free NH2 free amine group, and the C-terminal end will have a carboxyl group, isn't it? So as we know that the amino acid residues has a hydrogen group. Um, it has a side chain or a R group, which can be either hydrogen or there can be methyl group. There can be different types of so this R group will decide what type of amino acid it will have, and it has an alpha carboxyl group and an alpha amino group. Okay, so that is why the N terminal end has a free amine group and the C terminal has a free carboxyl group. So this free amino terminal or this free amine group which is present at the N terminal end can be utilized for sequencing a protein. So how is that done? So basically, this protein is first labeled with this compound, 1-fluoro-2,4-dinitrobenzene. So this 1-fluoro-2,4-dinitrobenzene can form a bond with this amino group which is present in the N-terminal end, and then through a process of Edman degradation, we can sequence our protein. So in the next slide, we will understand how this Edman degradation is done with the help of a figure. But what is Edman degradation? So this Edman degradation procedure labels and removes only the amino terminal residue from a peptide. So what happens is that this compound FDNB, it reacts with the N-terminal amino acid residue. Okay, so N-terminal amino acid residue is the amino acid which is present to or we can say it is the first amino acid residue of a protein. So the first amino acid residue of the protein will have a free amino terminal, and this amino terminal will react with the FDNB and form a compound. And by changing the pH, the first amino, so one by one, what happens? One by one, these amino acid residues can be cleaved, and this cycle we can we can have multiple cycles of this and ultimately sequence the protein. So let's understand through this figure. Okay, so you have your so this figure is taken from the book Leninger. This is a classical biochemistry book, and where you can see here, so you have your protein, and your protein will have a okay. So look at this figure. So this is the first amino acid residue of your protein. Okay, it has a free amino terminal end, which is the N-terminal end of the protein. Now, this free amino terminal end reacts with this phenyl isothiocyanate. Now, this first amino acid on reaction with this phenyl isothiocyanate forms this derivative. So, this derivative is called the phenyl thiocarbamyl derivative. Okay. Now, what happened? Now, the pH is lowered. So once this pH is lowered, what happens is this chemical reaction takes place. You can see here that this bond breaks and it will form this thiazolinone derivative of amino acid residue. So what you are getting over here, so do not you do not have to go understand the chemical reaction that is taking place, but just you have to understand the fact that this as a result of lowering the pH, what is happening is your first amino acid residue is forming a complex with this phenyl isothiocyanate, and this bond is getting cleaved. Okay, so what you are getting is this second amino acid residue that is so one of your amino acid residues got cleaved, and now you're left with the protein sequence which is starting from your second amino acid residue and continuing further. Okay, now once you now again you lower the pH, and you get this first amino acid residue, phenylthiohydantoin derivative of amino acid residue. So this cycle is repeated multiple times. So you can read this. The peptide bond nearest to the amino terminus of the protein or polypeptide is cleaved in two steps. So first, in the first step, what is done? A basic condition is a basic condition is applied. So in this basic condition, that is at high pH, this phenyl isothiocyanate will form bond with this amino terminus of the protein, and in the next step the pH is reduced. Now once the pH is reduced, this bond will be cleaved, and your first amino acid residue will be form a derivative with this phenyl isothiocyanate, and you will get the remaining peptide that is there. Okay, so now you can identify the amino terminal residue of the polypeptide, and you can once you are After multiple rounds of this process, you will get number of these. So now it is R1. In the next cycle, what you will have? You will have R2. Then the third cycle, you will get the R3, that is forming a complex with this compound. So you will have multiple species when this cycle when you are having multiple cycles of this process. So by through the series of cycle, you will have multiple amino terminal residues, and then you can identify the amino terminal residues of the polypeptide. Okay, so now let's go through this one, this theory, so that you can understand better. So the Edman degradation procedure, what it is doing. So remember the compound that is used for Edman degradation. It is 1-fluoro-2,4-dinitrobenzene. Okay, and this Edman degradation procedure labels and removes only the amino terminal residue. So in each cycle, it is removing one of the amino terminal residue from a peptide, leaving all other peptide bonds intact. Okay. Now this peptide is reacted with phenyl isothiocyanate under mildly alkaline condition, which converts the amino terminal amino acid to a phenyl thiocarbamoyl adduct. The peptide bond next to the PTC adduct is then cleaved in a step carried out in an anhydrous trifluoroacetic acid with the removal of the amino terminal amino acid as an anilino thiazolinone derivative. So this step actually, this low pH. This low pH is provided by this trifluoroacetic acid. So when this trifluoroacetic acid is added, it lowers the pH, and as a result, the second amino acid residue that is present, that is cleaved, the peptide bond is cleaved, and now you are having this free amino terminal of the second amino acid residue. Okay. And the first amino acid residue is removed in the form of this derivative, this anilino-thiazolinone. So this one that I was showing in this figure. Okay. Now the derivatized amino acid is extracted with organic solvents, converted to more stable phenyl thiohydantoin derivative by treatment with aqueous acid, and then identified. This use of sequential reactions carried out under first basic and then acidic conditions provides a means of controlling the entire process. So in each cycle, you will have one of these derivatives that will be produced. This one, this phenyl thiocarbamoyl adduct. This phenyl thiocarbamoyl adduct will have the amino acid residue, and and then you can extract this using these organic solvents. So so there is a multiple chemical reaction that is taking place. So you will extract with the organic solvents, and then you can convert to more stable this derivative. Okay. So this one. So once again, you lower the pH, and this is a more stable derivative, and you can sequence this and identify each amino acid. Okay. So each reaction with the amino terminal amino acid can go essentially to completion without affecting any of the other peptide bonds in the peptide or in the protein. And the process is repeated until typically as many as 40 sequential amino acid residues are identified, and the reactions of the Edman degradation have been so the reactions, they have been automated. Okay. So one by one, what will happen? So there will be 40 such cycles, and yeah, 40 such cycles will give you 40 different amino acid residues one by one, and this can be sequenced. So there are methods by which you can identify the amino terminal residue of the polypeptide. Now there are certain proteins which are having disulfide bonds. Now proteins which have disulfide bonds, the or proteins which have cysteine residues in their amino acid sequence, they can form intra disulfide bonds. So for example, there is a cysteine. So there is a protein in which there is a cysteine residue over here. So if you remember the structure of insulin, then you must be knowing that what happens is that there are multiple cysteine residues in insulin, and one of the cysteine residue can form covalent bond with the cysteine residue which is present in the with the other cysteine residue. So there is a disulfide bond which covalently links one region of the protein with the other region of the protein and folds the protein. Okay. Uh, so uh, so if you want to sequence the protein, what you have to do, you have to cleave this disulfide bond so that you can get a linear peptide. So once you get a linear peptide, then you can treat this peptide with different enzymes. You can treat it with trypsin, chymotrypsin. So each of these enzymes uh, have particular residues or they recognize particular residues and cut them. So some proteins cleave the peptide bond at the C terminal end of arginine or lysine. Some can cleave after cysteine. So there are different types of enzymes which we will learn in the upcoming slides. Okay. So enzymes called proteases catalyze the hydrolytic cleavage of the peptide bonds. And some there are some proteases which cleave only the peptide bond adjacent to particular amino acid residue. So what here you have a folded protein, okay? And this folded protein, the protein is folded due to the hydrophobic interaction as well as the covalent interactions. So these covalent interactions are formed by the disulfide bonds. So to cleave the disulfide bonds, what you have to do, you have to treat the protein with a reducing agent. So once you treat the protein with a reducing agent, what will it do? So the reducing agent will attach hydrogen bonds to the sulfur atom and this bond, this disulfide bond will break. Okay, so let's understand through this figure. So this is again from the book Leninger. So this is a protein. So for example, this maybe just for example, this can be a 30 this can be the 30 or 32nd amino acid residue, and this can be for, say, for example, the 20th amino acid residue. Okay. Now the 20th amino acid residue has a cysteine residue, and the 32th amino acid residue can also have a cysteine residue. Now this cysteine and this cysteine can form intra disulfide bond within the protein molecule. Now, when this disulfide bond is treated, it's treated with dithiothrietol, so DTT. Basically, when you treat this protein with DTT, what happens? This disulfide bonds get reduced, and once this disulfide bond is reduced, the this SS covalent bond, okay, disulfide bond will break. And now, what can happen is that when once this sulfur or this thiol group is reduced or this disulfide bond is reduced, you now you have SH. So this SH can again reform the disulfide bond. So this molecule is treated with iodoacetamide or iodoacetate. So what this iodoacetate does is that it forms this CH2COO minus group. It attaches this CH2COO minus group to the sulfur atom so that the disulfide bond is not reformed. Okay, is this point clear? Or other than this, you can also treat. So there are two methods by which you can cleave the disulfide bond. Either you treat it with reducing agents. So what are the reducing agents? So there are two common reducing agents which are used in lab. One is the beta mercaptoethanol and another is the DTT. So when you treat the protein with this reducing agents, your reducing agent will cleave the disulfide bond, or it will add hydrogen group or proton to this sulfur atom, and as a result your disulfide bond will break. Now to ensure that there is not that there is no there is no reformation of this disulfide bond, you treat it with iodoacetamide or iodoacetate, and this iodoacetate attaches this CH2COO minus group to the sulfur atom so that there is no reformation of the disulfide bond. And another method by which you can cleave the disulfide bond is by treating with performic acid. So basically this performic acid causes oxidation of the cysteine of this cysteine residues. So you get this CH2SOO or SO3 minus. Okay. So you can either cleave the, remember that you can either cleave the disulfide bond by treating it with reducing agent, or you can also perform oxidation by treating it with performic acid. Okay, so these are the two common methods illustrated. So oxidation of cysteine residues with performic acid produces two cystic acid residues. So these residues that are formed on treating with performic acid, these are now called the cystic acid residues, because you see this is the sulfate or SO3 minus group is attached to this CH2 to this CH2 group. Okay, or you can treat it with dithiothiazole or beta mercaptoethanol, which to form the cysteine residues. Um, to form this cysteine residues must be followed by further modification of the reactive. So this SH group is highly reactive, okay, and it can lead to reformation of the disulfide bond. So you do a carboxymethylation. So this is carboxymethyl. So CH2CO minus group getting attached to the sulfur atom is called carboxymethylation. So you have CH2 group and you have a CO minus group, and it will prevent the reformation of the disulfide bond. Okay. So a few chemical reagents also cleave the peptide bond adjacent to specific residues. So now, for example, you have lysine, then you have valine, then alanine, you have some tryptophan residue, then you can have an arginine residue. So when you treat your peptide, when you treat your peptide with particular enzymes. So you can either treat it with trypsin, you can treat with chymotrypsin, you can treat with endoproteinase lysine C, cyanogen bromide. So each of these enzymes have a particular residue which they recognize and cleaves the peptide molecule at that particular residue. Okay, so among proteases, the digestive, now trypsin is the most common enzyme which is used for cleaving your peptide. Okay, so what does trypsin do? So remember that trypsin will cleave at the car. So it will cleave after the lysine and the or the arginine residue. Okay. Among the proteases, the digestive enzyme trypsin catalyzes the hydrolysis of only those peptide bonds in which the carbonyl group is contributed by either a lysine or an arginine residue, regardless of the length of amino acid sequence of the chain. So it will cleave at the C terminal end of the lysine or arginine. So for example, consider you have some methionine group and you have this sequence leucine. Okay, you have a leucine. So now what will happen if you treat this particular peptide with trypsin? How many peptide fragments will you get? So if you treat it with trypsin, so trypsin, what will it do? It will cleave at the C terminal end of lysine or arginine residue. So it will cleave here. Okay, it will cleave. It will cleave here. Okay. So how many fragments you are getting? So you will get this over there. You will get one fragment. You will get two fragment, and there will be certain peptide molecules which will be having this entire peptide sequence. Okay, methionine, lysine, valine, alanine, tryptophan, arginine. So you can have, and you will have another fragment over here. Okay, so in total you will have four different fragments. Is it clear? Okay, so let's understand how this trypsin will cleave. So let's take an example of a peptide. So for example, you have a peptide which is having this sequence. There is methionine, then there is phenylalanine, then there is, for example, lysine, then alanine, then you have some leucine residue, then you have a cysteine residue, then you have arginine here. Okay, then you have valine residue, and then you have the lysine residue again here. And you have a tyrosine residue here. Okay. So you have a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You have a 10 amino acid peptide sequence. Okay. So you have 10 amino acid peptide sequence. So what will happen when you treat this peptide with trypsin? How many peptide fragments will you get if you treat this peptide with trypsin? So what will trypsin do? It will cleave at this peptide bond. So it cleaves at the C terminal end of lysine, means it will cleave here, it will cleave here, and it will also cleave here at this residue. Okay, so yeah, there will be some peptide fragments which will have this sequence: methylalanine, methionine, phenylalanine, and lysine. There will be some peptide fragments which will have this sequence: methionine, phenylalanine, lysine, alanine, leucine, cysteine, arginine. Okay, then you will get some peptide sequences which will have, or you will have a peptide which will have this sequence: alanine, leucine, cysteine, arginine. Okay, then you can have a peptide which is having this sequence: valine, lysine. Okay, so you can have multiple peptide sequences. Okay, now once you have these different peptide sequences, you can do a sequencing and then you can align them and determine the entire sequence of your peptide molecule. You can also have some peptides which are having this sequence. So starting from methionine, phenylalanine, lysine, alanine till here. So some peptides will have this sequence as well. Okay, so for so here they have taken an example of a four amino acid peptide chain. So for example, if you have four amino acid peptide chain, then what will happen? You have four small peptides with three lysine or arginine residues. Then how many peptide fragments will you get? So let's remove this one. Okay. Let's take an example of four amino acid peptide. So the four amino acid peptide with three lysine or arginine residues. Okay. So four amino, so there is methionine, lysine. So this is first, second, arginine, and then you have lysine. Okay. This is your four amino acid peptide. Now, if you treat this peptide with trypsin, how many peptide fragments will you get? So your trypsin will cleave at this position, it will cleave at this position, or it will cleave at, so they have this CO minus group, it will cleave at this portion as well. Okay. So you will get, so the first peptide fragment that you get will have methionine and lysine. So you will get a dipeptide. Methionine lysine. The second fragment that you get will have a sequence which is only having arginine. Okay, there will be a peptide fragment with the sequence methionine, lysine, arginine. Okay, and you will get a peptide sequence which is having all the four amino acid residues: methionine, lysine, arginine, arginine. So basically, when you are treating a four amino acid peptide which is having three lysine arginine residues with trypsin, you will have four different peptide fragments. Okay. So that is what is explained over here. Okay. Now the choice of a reagent to cleave the protein into smaller peptide can be aided by first determining the amino acid content of the entire protein, employing acid to reduce the protein to its constituent amino acid, and so if you know the sequence of your protein, so mainly this is required when you are doing when you want to do some mass spectrometry experiments. So in mass spectrometry experiments, your protein is first converted is the first you reduce the protein by treating it with with DDT. Then you treat it with these different enzymes like trypsin, and you get different peptide fragments. Now once you have got different peptide fragments, then you can do either then you can go for the further steps of mass spectrometry. We will understand how this mass spectrometry is done in a separate class, not today. This trypsin would be only on proteins that have an appropriate number of lysine or arginine residues. So that is why you must know the sequence of the amino acid residues in your protein. So if your protein does not have any lysine or arginine residues and you are treating this protein with trypsin, then what will happen? Then you will not get any peptide fragments. Okay. So then in that case, you have to use you have to use other enzymes other than trypsin. You can use other enzymes depending on what is the amino acid sequence that is present in your protein. Okay. So yeah. So now this was is from the lecture slide. So what are the different steps that are followed for sequencing of your protein? So basically sequencing of your protein means what you want to understand that what are the amino acid residues in your protein sequence. Okay, so what is done? So first of all, you have a folded protein. You want to convert this folded protein into a linear protein. Okay, so or at with this treating just with by treating with DTT, you will not get a linear protein, but these covalent bonds will be broken. So the covalent bonds which are formed by the disulfide bridges, these will be broken. So you can either treat your protein with the reducing agents like DTT or beta mercaptoethanol, or you can treat it with some oxidizing agents like performic acid, which results in formation of the cystic acid residues. Okay. So now once your peptide or once your protein is cleaved at this disulfide bonds, then you get the free SH groups. These are reactive. So to prevent the reformation of disulfide bond, you treat it with iodoacetate and you get this carboxymethylated cysteine residue. So once your disulfide bonds are reduced, the next step is what? Next step is to cleave the polypeptide chain. So how can you cleave the polypeptide chain? So you can treat it with proteases. Okay. So what are proteases? So these are chemical agents like so proteases means what? They are agents which will cleave your protein. Okay, it will digest the protein, and these are chemical agents that target proteins and have a specific recognition sequence, and they cleave after a particular amino acid. So these are basically chemical agents. So like you see this table here. So trypsin will always cleave after the lysine or arginine residue. Chymotrypsin cleaves after the aromatic residues. So you have to remember some of the not restriction sites, but some of the restriction sites we say for restriction enzyme, but for and these enzymes, they will cleave after particular amino acid residues. So chymotrypsin will cleave after aromatic amino acid residues after phenylalanine, tryptophan, tyrosine. Pepsin usually cleaves after leucine, phenylalanine, tryptophan, tyrosine, and cyanogen bromide will cleave after methionine. Okay, what are the different methods by which you can sequence your protein? So your first method is that you can treat your protein with FDNB. So if you treat your protein with FDNB, what we just now discussed, the Edman degradation steps. So when you treat with FDNB, then the free amino terminal end of your protein, this free NH3 plus group, which is the first amino acid. So the first amino acid will have a free amino terminal group. So this free amino terminal group will react with FDNB and it forms this derivative, which can be detected and its amino acid can be analyzed. Or what you can do, you can treat this protein with disulfide, or you can treat this with reducing agents. So once you treat with reducing agents, you get SH groups. And now this protein is then treated with trypsin. So once you treat it with trypsin, you will get different peptide fragments. So these are the different peptide fragments that you have obtained. So you see that when your protein is being treated with trypsin, you are having your protein, your peptide molecule is ending in either lysine or arginine. Okay, either lysine or arginine. So you have this GASMALIK. So this is glycine, alanine, serine, methionine, alanine, leucine, isoleucine, and lysine residues. You will get a peptide fragment with this amino acid sequence. You will get a peptide sequence which may be present between this peptide sequence and this peptide sequence. Okay. So you will get in between this. Okay, you will get other sequences which will have this particular amino acid sequence. So how you can establish the amino acid sequence in your protein? So you treat your protein with trypsin. You get four different peptide fragments: T1, T2, T3, T4. You treat it with cyanogen bromide. Again, you get on treating it with cyanogen bromide, you are getting three different peptide sequences. Now, once you have got these different peptide sequences, you can sequence these peptides by Edman degradation method. And once you sequence once you know the sequence of each of these peptide fragments, then you can align them and you can get the complete sequence of your protein. Is it clear? So, so the idea is that when you are treating with cyanogen bromide, you are getting this sequence EG starting with E and ending in methionine. Okay. And when you are treating with trypsin, you are getting a sequence as the which goes like this: EGA, Y, HD, FE, PID, PR, proline and arginine. So now this sequence and this sequence, if you match, then you will get to know that after this arginine, you have this glycine, alanine, serine, and methionine residue. Okay. Now this sequence that you have peptide that you have obtained, TKDCV, HSD, this you can align with this one, right? So you can align these different peptide sequences and you can get the complete sequence of your protein. So this particular sequence which I have obtained on treating with trypsin, this you can see this is ending with TK and this is starting with TK. So obviously what will happen that this after this threonine and lysine residue, we will have this aspartate and these different amino acid residues. And here you can see that this has this particular sequence, you can align with this one, this sequence. So you align all these three and you can establish these sequences. Okay, you can write, you can have, so if you want to understand how this is happening, you can write all the sequences in your notebook and you can try to establish the entire sequence and you will understand that how this entire sequence. So basically you are doing an alignment and then you can by aligning this sequence you can get the complete protein sequences. So you can do this on your own and you can then you will understand that how we can analyze that, which is the initial peptide sequence which is which will come in the middle, which will come in the end. Okay. Yeah, so these different. So now what you have your protein, you have treated it with disulfide, you have treated it with some reducing agents, DTT. So DTT has resulted in breaking of this disulfide bridges. Now after that, you have treated with some enzyme like trypsin. Okay. And now you are having different peptide sequences. Okay. You are having different peptide fragments. Now each of these peptide fragments, you can get the sequence of each of these peptide fragments by doing an Edman degradation. So this peptide fragment that you have, you treat it with DNFB, and you will, after several cycles, you will get to know the sequence of this peptide fragment. You have this peptide, they have the second peptide fragment, you again treat it with DNFB, and you get the sequence of this second peptide fragment, and one by one, you can get the sequence of each of these peptide fragments. And then you can align these sequences and get the sequence of your entire protein. Okay, so that is what has been shown in this figure as well. So once the peptide fragments are generated, we can start sequencing of each polypeptide chain. So for sequencing of each polypeptide chain, you can go for the Edman degradation. So what Edman degradation does, it will identify the N-terminal residues one by one. So the N-terminal amino acid analysis is a three-step process. So in the first step, what will happen? So we discussed already what how this process is taking place. So when you treat your peptide with this DNFB will form a you can either treat it with DFNB, which is 1-fluoro, 2,4-dinitrobenzene, or you can also treat it with dancing chloride. So these chemical reagents, they are very specific, that in the sense that they will only react with the terminal amino acid. Okay, so one by one, each of your amino terminal residue will form bonds with this with this chemical reagents, and then you can identify the amino terminus, one each of these amino acid residues which are present in your N-terminal end. Okay, these reagents are also label-free amino groups present on basic amino acids such as lysine and arginine. So yeah, so the disadvantage of this is that your lysine and arginine amino acid, if you see, they have your lysine and arginine residues have free amino group in their side chains. So the R group, which is present in arginine, and the R group, which is present on lysine, they have the free amino acid side amino. They have a free amino terminals in their side chain. So these free amino terminals can also react with this this dancyl chloride and or DNFB. Okay. So this difluoro, so remember this dinitrofluorobenzene reacts with the free amine group to form a dinitrophenyl amino acid complex. So this is the complex, dinitrophenyl amino acid complex. So this is the first amino acid, R1, you can see here. This R1 will form a this dinitrophenyl amino acid complex, and you will once you treat it with an acid, you can get this second and third amino acid residue as well. Now, in the next step, you hydrolyze the protein. So first you treat it, you react the protein with DNFB, and you get and your amino acid, amino terminal amino acid will form a bond with this DNFB. Okay, then what you do, you lower the pH as I was mentioning. First, you increase the pH, then you lower the pH. So when you lower the pH, there will be hydrolysis of this dinitrophenyl amino acid complex, and it leads to the breaking of the peptide bond to release the dinitrophenyl amino acid complex in solution. So once you lower the pH, then what you are getting this compound. So this is the this is the dinitrophenyl amino acid complex, and then you can separate these different amino terminal compounds that you have got by HPLC, by doing a HPLC or TLC. So there will be, you will get different so do you understand what I'm trying to say? So you will have, so each cycle will give you one of such amino acid complex, dinitrophenyl amino acid complex. So in the first cycle, you will get this dinitrophenyl amino acid complex. In the next cycle, you will have the R2. R2, the amino acid which is having the R2 side chain. In the third, you will get the amino acid which is having the R3 side chain. In the fourth, you will get the fourth amino acid which is complex to the which is bound to the fourth side chain. Okay, so in this way you will get different molecules, right? Different amino acid residues which will be which will form a complex with this dinitrophenyl compound, and then by HPLC or TLC you can separate each of these and you can sequence them. So comparing with the standard amino acid. So the standard amino acid has particular elution volume that is known, and then comparing those, you can assign that this peak 1 is corresponding to this amino acid, peak 2 this amino acid, peak 3 is corresponding to this amino acid, and by comparing with the standards, you can get the entire sequence of your protein. Okay. So this is the same thing which has been explained. So first, at high pH, alkaline pH, this one will form a complex. Okay, the same thing is written here. Okay. So first you get a phenyl thiocarbamyl derivative. and then under acidic condition, this derivative will be cleaved. So the terminal amino acid will be cleaved. You see here, this is the first amino acid that will be released. Now you are left with the second amino acid, which will now have the free amino terminal group. This will again react with this phenyl isothiocyanate, PIC. Okay. So in this way, you will get in each of the cycle, you will get one of the amino acid residues or the N-terminal end of the amino acid residue, which will form a complex with this. So you can treat the protein with carboxypeptidase to release the C-terminal amino acid and test the solution in a time-dependent manner. So your trypsin was cleaving at the or it was cleaving after the C-terminal end of lysine and arginine, isn't it? But if you treat it with carboxypeptidase, so carboxypeptidase will release the amino acids from the C-terminal end. What does this mean? So, for example, you have this molecule cysteine, methionine, lysine, serine, and say, for example, alanine. So, what carboxypeptidase does, it will start cleaving from this end. Okay. Carboxypeptidase will start cleaving. This peptidase react with the COO minus group, which is free at the C terminal end of the protein. So like your this phenyl isothiocyanate. Phenyl isothiocyanate was reacting with the amino free amino group which is present towards the N-terminal end of the protein. Carboxypeptidase will start cleaving the protein from your C-terminal end. So you will have sequence which goes like A, S, L. So in that way it is cleaving the protein. So is it clear? So it will release the amino acid from the C-terminal end of the protein. Okay. Now you can again sequence each of these. You can order the peptide fragments. How will you order? How will you do sequentially arrange these peptide fragments? So the usage of different protein cleavage reagents, like here you saw in this figure, we have used two different reagents. One is we have treated with trypsin and we have treated with cyanogen bromide. And we have the information that trypsin will cleave after the lysine and arginine residue. Cyanogen bromide will cleave after the methionine residue. Now you can, you are getting different peptide fragments. You are aligning these peptide, you are sequencing each of these peptide fragments, and then you are aligning these peptide fragments to get the entire sequence of the protein. So that is what is the same thing has been written here. You can treat your protein with different enzymes, and then you can sequence these peptide fragments, and then by aligning these peptide fragments, you will get the entire sequence of your protein. Okay. Now, locating the disulfide bonds, the protein cleavage by trypsin is performed with or without breaking disulfide linkage. Amino acid sequence analysis of the fragments will provide the site of disulfide bond. The presence of one disulfide will reduce two peptide fragments and will appear as a one large peptide fragment. Okay, and also you can sequence these peptide fragments by mass spectrometry method. Okay, so we will learn about this MALDI and ESI TOF. So MALDI TOF and the ESI electron spray ionization method that is done for sequencing the amino acid sequence in a protein. What are the steps in it? So by mass spectrometry method also you can characterize each fragments to deduce its amino acid sequence. So by using mass spectrometry also you can get the sequence of your protein. There's by using this tandem MSMS technique, or you can also get the information about the molecular weight of your protein. Okay. Also, there are different mass spectrometry based method. One which can give you the sequence of your protein, one which can give you the molecular weight of your protein, and how each of these techniques, each of the technique act or principle of this technique, we will have a separate discussion in another class. But now let's move on to spectroscopy. So what do you understand by spectroscopy? So what is spectroscopy basically? So spectroscopy is a technique where what we are doing, we are trying to understand the interaction of electromagnetic radiation with matter. We are trying to understand the interaction of electromagnetic radiation with matter. So how the interaction of electromagnetic radiation with matter will will provide us the information about the functional groups which are present in a molecule, about the kind of bonds which are present in a molecule. It can give us information about the length of the bonds which are present in a molecule. Okay, so it can give us different information about the characteristics of a molecule. Okay, so why is it so? Because if you see that these different the biomolecules that are present, they are having they can have a carbon-carbon single bond, they can have carbon double bond, or they can have different functional groups attached to them. Now, each of these functional groups are associated with a they have particular vibrational energies. They have particular rotational energy or rotational frequencies. So when this energy of the electromagnetic radiation will interact with this matter, it will cause the excitation of electrons from one orbital to the other orbital, and utilizing it, we can characterize the different properties of a of the of our molecule. So basically, when we talk about the electromagnetic radiation, it starts from the gamma rays and up to the radio waves. So um, we can have a we can have different energies of the electromagnetic radiation. So the gamma rays, so from here if we look, so the gamma rays are having the maximum energy, and as we go towards the right, the energy of the electromagnetic radiation decreases. So energy decreases, but the wavelength is, so energy is what? Energy is hc by lambda, isn't it? So from the left side of the electromagnetic radiation towards the right side, your energy is decreasing while your wavelength is increasing. Okay. In this in this electromagnetic spectrum, you we so this is the visible region of the electromagnetic spectrum. So this lies in the wavelength range starting from 400 nanometer to 780 nanometer. Uh, and other than this, lower wavelengths, as we move towards here, the wavelength increases. So you have, but your frequency increases. Okay. So, so what happens is the electrons are arranged in specific molecular orbitals. So when you excite your molecule with an electromagnetic radiation, this electron will absorb the energy of the electromagnetic radiation, and or it will absorb. So electromagnetic radiation is what? They are composed of these photons. So each of these photons has an energy. Each photon will have an energy h nu. So this energy h nu will be absorbed by the electron, and it will jump to the higher energy state. Now, when this will come back to its lower energy state, the energy that it releases, this energy can be in the range of, it can be in the range of visible region. It can be either in the range of UV region, or the energy that is released, it can be in the range of IR region. Okay, so so the so now by using detectors which can detect the energy which is released in the IR region, the energy which is released in the UV region, energy which is released in the visible region, we can get information about the different properties of a molecule. Okay. So we will understand this one by one. So when so your if you look at the structure of an atom, then inside an atom, the electrons are residing in different orbits or orbitals surrounding the nucleus, and yeah, and these electrons are will have discrete electronic energy levels. So these are also called as the orbits, are also called as the energy levels in the quantum mechanics term. We what we say that is there are a particular electron density where the electron can be where there is the maximum probability of finding the electron. So we do not say orbits or orbits, but what we say is that there is a particular density around the nucleus where the probability of finding the electron is maximum. So these are called the energy levels. So it can so these energy levels can also be called as orbitals. Right. When we excite this, so this is the same thing that you have explained. So the emission can be in different range of the electromagnetic radiation. So so as I was explaining that the molecule can have different transitions. It can have electronic transitions, it can have rotational, it can have nuclear transition, you can have vibrational transitions. Now each of these transitions are associated with the release of a particular energy of radiation. Okay. Uh, so vibrational transitions release energy in the IR region. Okay. Or this electronic transitions. For electronic transitions, you need to excite the molecule in the UV region. So we will look at this one by one. So the radio frequency waves. So where do we use radio frequency waves to excite our molecule? So the radio frequency waves are used for exciting our molecule with energies which are in the radio frequency range, and these frequencies are very have very low energy, right? They are having very low energy, and so it can be used for studying the energy differences in the nuclear and electronic spin states. Okay. Uh, without going into too much detail, that radio frequency will help in understanding the transitions which are taking place in the nuclear and electronic spin states, and these frequencies will therefore find application in the nuclear magnetic resonance and EPR spectroscopy, electron paramagnetic resonance spectrometry. Now, microwaves have energies between those of the radio frequency waves and the infrared waves, and find application in the rotational spectroscopy and electron paramagnetic resonance spectroscopy. Then there are infrared radiation. So infrared radiation can be used for vibrational spectroscopy for understanding for to analyze the functional groups which are present in a molecule. So infrared radiations are the energies associated with molecular vibrations, which fall in the infrared region of the electromagnetic spectrum. So infrared spectroscopy is therefore also known as the vibrational spectroscopy, and is a very useful technique for functional group identification in organic molecule. You can use the UV visible region for the electronic transitions. Okay, UV and visible regions are involved in the electronic transitions. So a molecule can be arranged in different orbitals, right? So this is N, this sigma star, sigma pi, and this is pi star. So these are the electronic energy levels in which an electron can be arranged. Okay. So when you apply an energy which falls in the UV region, UV visible region, then the molecule can transition from N to pi star. It can move from, depending on the energy, it can move to Now this one, pi to pi star transition can be possible, or there can be pi to sigma star or sigma to sigma star. So there can be different electronic transitions that are observed when you excite the molecule in the with energy which is lying in the UV visible region. Okay, so these are the electronic transition, pi to pi star, n to pi star, sigma to sigma star. These are the different electronic transitions that take place. So electronic transitions means what? Your electron is transitioning from one orbital to the other orbital. So this transition will take place when you excite the molecule with UV or visible light. And what is X-ray radiation? So X-ray radiations are having a very large energy. So they are high energy electromagnetic radiation and causes transitions in the internal electrons of the molecule. So the electrons which are residing in different orbitals, they can transition from one orbital to the other orbital when this X-ray radiation is applied. Okay, so as I was explaining the UV visible spectroscopy, so the UV visible spectroscopy means you are the energy that is being applied it falls in the UV or the visible region. So the UV region has a wavelength in the range of 10 nanometer to 400 nanometer. So UV region can again be divided into the far UV region or the near UV region. So near UV regions means the wavelength will be near to the visible region. So that is near UV wavelength. So the near UV wavelength has a range from 250 nanometer to 400 nanometer, and the far UV region wavelength will have a wavelength starting from 190 nanometer to 250 nanometer. Okay, and your visible region has energy which corresponds to the wavelength from 400 nanometer to 780 nanometer. So the absorption of UV and visible light is through the transition of an electron in the molecule from lower to a higher energy molecular orbital. So in chemistry, you must have learned about this concept HOMO and LUMO. So these different transitions that take place when you are applying, when you are exciting the molecule with radiation in the visible region. So it can have these transition. It can transition from N to pi star. The molecule can transition from pi to pi star. The molecule can transition from N to sigma star or sigma to sigma star. So the sigma to sigma star transition is a high energy process. So for transition from the sigma to sigma star, there is a high energy gap between the two. So you need to apply radiation which is in the low wavelength region. Okay. So if you want transition in the sigma to sigma star, then you have to excite the molecule in the vacuum UV region. Okay. So in the range of in the with a wavelength, energy having a wavelength less than 190 nanometer. Now alkanes, wherein only sigma to sigma star transition is possible through absorption bands around 150 nanometer wavelength. So alkanes can show transition in the sigma to sigma star. From sigma to sigma star, alkanes so they will have a lower wavelength in the far UV region. Okay. Now alkenes have pi and pi star orbitals, and they can show several transitions. So alkenes they are conjugated, right? They are having double bonds. So one is a pi bond and another is a sigma bond. So because of this, it can show different transitions. It can show transition from pi to pi star orbital. It can show transition from so the lowest energy transfer is this pi to pi star. Pi to pi star, that will give an absorption band of 170 to 190 for non-conjugated alkenes. And the presence of non-bonding electrons in a molecule, aliphatic ketone, for example, the absorption band around 185 nanometer rises due to the pi to pi star transition in the carbonyl group. So if a carbonyl group is present, it will show transition in the pi from pi to pi star and that gives an absorption band around 185 nanometer. If it is alkene, then it will have an absorption band in the 172 to 190 nanometer region. So now How so for UV visible spectroscopy, what you have to do, you have a sample, okay, which is placed in the cuvette, and you have a source of light. Now this source of light will produce an electromagnetic radiation, um, which will excite the molecules. Okay, and then the energy which is released will be detected by a detector. Okay, so this is a very simple um, schematic of how these UV visible spectrometers they work. So you have a source of light, which will so you can set the source of light. You can have different excitation wavelength. You can either this UV visible spectrophotometers that are there, they can excite your sample in the UV visible range. Okay, so your UV visible range will start from around 190 nanometer to 400 nanometer. Okay, so in this range of wavelength, you can excite your sample, and then you can detect the or you can your sample will absorb the light, and then the amount of light which passes will be detected. Okay. So the light enters the instrument through an entrance slit. So this is a visible light source, or you can have a UV light source. You can set of the wavelength at which you want the sample to absorb. Now the light will enter the instrument through an entrance slit. It is collimated and focused on the dispersing element, typically a diffraction grating. So this is the diffraction grating. Okay. So the light of the desire is selected simply by rotating the monochromator and impinged on the sample. So you have a monochromator. So you have a source of light. The mirror will reflect this source of light towards the monochromator. What your monochromator is doing? Now, a monochromator will select a particular wavelength of light. Okay, the light of desired wavelength is selected by simply rotating the monochromator. There are diffraction grating channels as well. So there are monochromator, and then the particular wavelength of light with the help of this monochromator, the sample can be radiated with a particular energy of light. Now, the intensity of the radiation transmitted through the sample is measured and converted to absorbance or transmittance. So you have a sample, you are radiating this sample with a particular wavelength of electromagnetic radiation or a particular wavelength which lies in the UV visible region, and the energy of light which will pass through this sample, that will be your transmittance. Okay. Now there you can have single beam spectrophotometer. So single beam spectrophotometer has only a um, single chamber where you can place your cuvette. While double beam spectrophotometer will have two different sample holding chambers. So in one of the chambers you need to add your buffer or the reference solution, and in other you will have the sample which is present in your buffer. Okay, so for protein, you will have a reference cell where you will add only the buffer in which your protein is present, and in another cuvette you will add the protein. Okay, and then you can manually subtract the absorption by the buffer, and you can get the absorption of the sample. Okay. So a dual beam spectrophotometer, it has two light beams. Okay, it has two light beams. One of which will pass through the sample, other will pass through a reference cell. Now this allows the reproducible measurement, as any fluctuation in the light source or instrument electronics appears in both reference and the sample, and therefore can easily be removed from the sample spectrum by subtracting the reference spectrum. So you get the spectra of the reference, and you get the spectra of the sample. Now you can subtract the spectra of the reference, and you can have only the spectra that is produced by your sample. Okay. Monochromator in this spectrophotometers is placed after the sample. Okay. After the sample, so that the sample is exposed to the entire spectrum of the incident radiation, and the transmitted radiation is dispersed into its components. Now, this UV visible spectroscopy, it follows the principle of Beer-Lambert's law. So what is the Beer-Lambert's law? It says that the absorption of a sample is directly proportional to the concentration of the sample. The more the concentration of the sample, the more will be the absorption. Okay, so what is the equation? That is absorbance equal to epsilon Cl, where epsilon is what? It is the molar extinction coefficient, C is the concentration of the sample, and L is the path length. So absorption by a sample will is directly proportional to the length of the is directly proportional to the length of the cuvette. Okay, basically, if you are using a 10 millimeter cuvette, then the path length, the path length of the cuvette is what? It is 10 millimeter. It will depend on the concentration of the sample, and it will also depend on the molar extinction coefficient. So it is quite intuitive that at higher concentration of the absorbing species in a sample would lead to higher absorption of light. So if the concentration of the sample is higher, then the absorption will also be higher, and the higher the thickness of the sample should result in higher absorption. So the equality showing linear relationship between absorbance and concentration is known as Beer's-Lambert law. So you can either express this in this form, or you can also express it in the form of transmittance. So absorbance is what? Absorbance is log I by I naught, or absorbance is log 1 by T. Log 1 by T. And transmittance is what? Transmittance is basically the ratio of transmittance is the ratio of the amount of light that is transmitted, or that is transmitted through the specimen divided by the amount of light which is falling on the specimen. So transmittance is basically I by I naught. So basically you have a sample, you excite it with a particular intensity. So that is I or I naught. And so once you irradiate with a particular intensity of light, some of this intensity of light will be absorbed by the sample, and some of the intensity will be transmitted. So I is the intensity of the transmitted light, and I naught is the intensity of light which is incident on the sample. So transmittance is the ratio of I and I naught. Okay, so now the relationship between absorbance and transmittance is what? It is absorbance equal to log 1 by transmittance. Okay, so or you can write it that absorbance is equal to the negative log of transmittance. Okay. So if you bring this transmittance to the numerator, then you will have a negative log sign, isn't it? So absorbance is the negative logarithm of transmittance. Okay. So on transmittance is another way. So either you can express it in the form of absorbance, or you can express it in the form of transmittance. The amount of light which is reaching the detector, that is your transmittance. So it is another way. So so that the transmittance and absorbance, they are somewhat inversely related. But the negative log in the negative log. So the more the absorbance, the less will be the transmittance, isn't it? So that is why it is absorbance is minus log of the transmittance. So it's simply the ratio of the intensity of radiation transmitted to the sample, that is I, to the that of the incident radiation. Incident radiation is I naught. As it is clear from the definition of absorbance and transmittance, they are both dimensionless quantities, because it is the ratio of the intensity of light which is transmitted to the sample, and the incident light which is falling on the sample. So this will be, both are intensities. So this is a dimensionless quantity. So as a result, this transmittance is expressed in arbitrary units. The unit, it will have no unit, right? So that is why it is expressed as EU. Okay. So you can take an absorption spectra of your sample. So for example, here the absorption spectrum of N-acetyl tryptophan. tryptophanamide has been shown. So if you take the absorption spectra, you can see that starting from 200 nanometer to 400 nanometer, you can get that at which wavelength your molecule is absorbing maximum. So the quantity of interest in an absorption spectrum is the molar absorption coefficient. So what is molar absorption coefficient? So this molar absorption coefficient will vary with the wavelength. Okay, the wavelength at which highest molar absorption coefficient is observed is represented as lambda max. Now there are certain cases where you can get deviations from the Beer-Lambert law. So the Beer-Lambert law can be used to determine the epsilon values. Okay, so depending on where you are having the maximum wave absorption. So the wavelength which you are getting the maximum absorption will be a lambda max, and this lambda max will correspond to a particular molar ellipticity or molar extinction coefficient. Now, alternatively, the knowledge of this molar extinction coefficient enables the user to calculate the concentration of a compound in a given solution. It is, however, not uncommon to observe deviation from the Beer-Lambert's law. So when you are, for example, you are measuring the absorption of a protein at 280 nanometer, then you need to know that what is the molar extinction coefficient of the protein at 280 nanometer. So the molar extinction coefficient of the protein at 280 nanometer will depend on the amount of light which is absorbed by the tryptophan residue at 280 nanometer, the tyrosine residues at 280 nanometer, the phenylalanine, the cysteine. So all these will give you the particular molar extinction coefficient at 280 nanometer. Okay, so utilizing that, you can get the absorbance at 280 nanometer, and so each protein has a particular molar extinction coefficient, and utilizing that, so that molar extinction coefficient will depend on the tryptophan and the tyrosine residues which are present in your protein. And once you have the information about the molar extinction coefficient, you can calculate the concentration of your protein using this Beer-Lambert's law. Okay, so the Beer-Lambert's law generally holds good only for dilute solution. At higher concentration, the molecules come in close proximity, thereby influencing their electronic properties. Although introduced as a constant at a particular wavelength for a compound, this molar extinction coefficient depends on the concentration of the compound and therefore results in deviation from linearity. At low concentration, however, epsilon can be practically assumed to be constant. So if a molecule undergoes a chemical reaction and the spectroscopic properties of the reacted and unreacted molecular species differ, there can be deviation from Beer-Lambert's law, or there can be instrumental factors. So as epsilon is a function of wavelength. So if there are some instrumental factors where the sample is not getting excited with or not having that particular wavelength for absorption, then also this Beer-Lambert's law will show deviation. So use of polychromatic radiation will result in deviation for linearity between absorbance and concentration. So for practical purposes, the sample giving absorbance values between 0.05 to zero point 0.5 are considered highly reliable. At lower concentrations, the signal to noise is small, while at higher concentration, absorbance values underestimate the concentration of the compound. Okay, so for practical purposes, this absorbance, which falls in the range of 0.05 to zero point 0.5, is considered reliable. Okay. So you need to accordingly adjust your concentration. So if your concentration is very high, then you will get an absorbance which is near to 1. So so this absorbance that you get is not reliable. So what you have to do, you have to dilute your sample and then you have to take the absorbance. So what is usually done is your sample should have an absorbance in the range of 0.2 to zero point 0.8. If it is lying above this range or if it is lying below this range, then the absorbance value that you are getting, that is not reliable, and you need to accordingly dilute it so that you get an absorbance value in this range. And then when the sample is having an absorbance in this range, then you can use that for calculating your concentration. Now what can happen So there can be factors that could shift the absorption spectra to smaller wavelengths or can increase or decrease the absorption intensity. Okay, so before understanding how conjugation causes shift in the absorption spectra, let us look at some important terms that are used to refer to the shifts in absorption spectra. So there can be the molecule can show changes in the absorption spectra. So depending, so for example, if a protein is interacting with a particular ligand, then you're depending on depending on the concentration of the ligand or the where this ligand is interacting with the protein, your protein can show four different phenomena. It can either show a hypochromic shift, hyperchromic shift, hypochromic shift, bathochromic shift, and hypsochromic shift. So if there is some interaction and you're on interaction with that particular molecule, your absorption spectra is shifting towards the right side. So that is called a bathochromic shift. If it is shifting to the left side, it is called a hypsochromic shift. If the absorption is increasing, that is a hyperchromic shift. If the absorption is decreasing, that is a hypochromic shift. Okay. So, a shift of the absorption spectra towards smaller wavelength is a hypsochromic shift. Shift towards longer wavelength is bathochromic shift, and shift to or increase in absorption will be called hyperchromic and decrease in absorption will be called hypochromic. So there can be chemical groups that are attached on a molecule. These chemical groups are called oxochromes. So oxochromes are chemical groups that result in a bathochromic shift. Bathochromic shift means there will be shift in the absorption spectrum towards higher wavelength. Okay. The strongest oxochrome are the OH, NH2, OR, etc., which possess non-bonding electrons. So because of the non-bonding electrons, when these molecules, when these groups are present in your molecule, they will show a bathochromic shift. Okay. Or the oxochrome modified chromophore is a new chromophore in real sense. The term oxochrome is therefore rarely used these days, and the entire group can be considered as a chromophore different from the basic chromophore. So, depending on the conjugation that is present in a molecule, depending on what type of functional group which is present in the molecule, it can show different behavior. It can show these four different shifts in the absorption spectrum. Now, the solvents which you are using for spectroscopic methods, they also have a particular absorption. So, for example, this water absorbs at 190 nanometer. Acetonitrile also absorbs at 190 nanometer. Cyclohexane, methanol, 95% ethanol. So they all have a particular absorption. So always what you have to do when you are dissolving your sample in these solvents, you need to take the spectra of the solvent, you need to take the spectra of the sample, and then you have to subtract the amount of absorbed light that your sample is absorbing and the amount of light that your solvent is absorbing. So only then you can get the amount of light which is only being absorbed by the sample and not by the buffer or not by by the solvent in which your sample is present. Okay. So the amino acids and proteins they are having basically they have three chromophores. Okay. One is the peptide bond. Second is your is the aromatic side chains or aromatic amino acid residues. Aromatic amino acid residues, and third is what? Basically your cysteine. So these are the chromophores which are present in a protein molecule. So amino acids and proteins are among the 20 amino acids that constitute the protein, the tryptophan, tyrosine, and phenylalanine. They absorb in the near UV region. So they absorb in the 280 nanometer wavelength of UV light, UV radiation. Okay. So the absorption maximum of phenylalanine is 200 per mole per molar per centimeter at 250 nanometer. So phenylalanine shows maximum absorbance at 250 nanometer. Tryptophan shows maximum absorption at 270 at 280 nanometer, and tyrosine shows maximum absorption at 274 nanometer. Okay, now these disulfide linkages formed through oxidation of cysteine residues, they also show absorbance in the near UV region with a weak epsilon maxima of 300, around 250 to 270 nanometer. So the absorption spectra of proteins are therefore largely dominated by the tyrosine and tryptophan in the near UV region. In the far UV region, the peptide bond emerges as the most important chromophore in the proteins. So the side chains of aspartic acid, glutamic acid, asparagine, glutamine, arginine, histidine also contribute to the absorbance in the far UV region. So remember that the peptide bond that is present in your protein, that will absorb in the far UV region. So they absorb in around 190 nanometer wavelength. Your tryptophan, tyrosine, they will absorb around 280 nanometer wavelength of light, and the cysteine will absorb in the range of 250 to 270. So the main chromophore are actually the tryptophan and tyrosine. So the absorption spectra of proteins will therefore be largely dominated by the tyrosine and tryptophan, which is present in the near UV region. Okay. So now nucleic acids have these nitrogenous bases, purines and pyrimidines, and because these nitrogen bases also can also absorb. Uh, so uh, so the nucleic acid absorb very strongly in the far and near UV regions in the electromagnetic spectrum. The absorption is largely due to the nitrogenous bases. That is what I was saying, that the nitrogenous bases will show absorption. So the molar absorption coefficient vary for the nucleotides at 260 nanometer. The average epsilon max can be taken as 10 to the power 4 per mole percent. So for proteins, the absorption is taken at which wavelength? 280 nanometer. For nucleic acid, your absorbance is taken at which wavelength? It is taken at 260 nanometer, because the purine and pyrimidine nitrogenous bases, they absorb strongly in the 260 nanometer wavelength of light, while the protein shows strong absorption at the 280 nanometer wavelength of light. So it is important to mention that the nucleotides show hyperchromicity when exposed to aqueous environment. The absorbance of free nucleotide is higher than that of single-stranded nucleic acid, which is higher than that of double-stranded nucleic acid. So if you have free nucleotides, and you are measuring the absorbance at 260 nanometer, then which one will give the maximum absorbance? Your free nucleotides will give the maximum absorbance, followed by the single-stranded DNA molecule, and this will be followed by the double-stranded DNA molecule. Yep. There can be other chromophores present like NADH, NADPH, FMN, FAD, or there can be porphyrins such as heme, chlorophyll, plant pigment, and all these different chromophores also show particular absorption in the UV and visible region. Okay, so now answer this question. So the relationship between absorption and transmittance is what? So they follow a negative logarithm, isn't it? They follow a negative logarithm relationship. So your absorbance is epsilon CL. This is the product of this, depends on the molar extinction coefficient, the concentration of the sample, and the path length. And you can also express absorbance in the form of transmittance. So absorbance is negative logarithm of transmittance. Okay, and what is transmittance? Transmittance is the ratio of the transmitted light to the incident light. You can also write it in this form, minus log I by I naught, or you can write it as log I naught by I. Okay. So the answer is, it is negative logarithm of transmittance. So moving to the next question, what is a collimator? Why is collimator used in monochromator? So if you see this figure, this one. So this figure they have not shown the collimator, but their collimator, what it is doing, it is basically answer is that it will focus parallel incident radiation onto dispersing device. Okay, answer is C. The collimator will help to focus the light at particular incident radiation onto the dispersing device. Okay. Now there are two compounds A and B which have molar absorptivity 1200 and 1500 mole per liter per centimeter respectively. So now if we have two compounds A and B, one is having a molar ellipticity or molar extinction coefficient at 1200 and another is having a molar extinction coefficient at 1500, then which one will absorb more? Obviously the one which is having more epsilon value will absorb more, because according to Beer-Lambert's law we know that absorbance is directly proportional to the molar extinction coefficient, concentration, and the path length. So the more the molar extinction coefficient, the more will be the absorbance. So the one which is, so your B molecule is having 1500 molar extinction coefficient, so it will show more absorbance. So that is why compound B can be detected at very low concentration, because its molar extinction coefficient is 1500. So it can be detected at low concentration than compound A. Okay. Now answer this question. So this is a numerical. So you have your path length is given. Path length is 2 centimeter. Your molar extinction coefficient is also given. It is 2 into 10 to the power 4 liter per mole per centimeter. So why does this have this molar extinction coefficient have this unit? Because if you do this, this absorbance equal to epsilon CL, so epsilon will be 1 by CL. So your concentration is mole per liter, right? And your path length is centimeter. So that is why the unit of epsilon will be what? It will be per mole liter per centimeter. This will be the unit. So you can solve this, right? So your absorbance value is 1. So if you put this in this equation, absorbance equal to epsilon CL. So your absorbance is 1. Your epsilon is 2 into 10 to the power 4. Your concentration is unknown. You need to find the value of the concentration. Your path length is 2 centimeter. Then from this equation, you can get the value of C. So your concentration will be 1 by 4 into 10 to the power 4. That is 0.25 into 10 to the power 4, or it will be 2.5 into 10 to the power minus 4. So it will be 2.5 into 10 to the power minus 5. The answer is actually option A. So in today's session, I did not discuss about the IR spectroscopy. I did not talk about the MALDI and the mass spectrometry techniques. In the next class, we will look into these techniques, and also we will discuss other part of the UV visible spectroscopy that is remaining. So I will end today's class here, and I'll again start the discussion in the next class. Thank you.